Your microphone was close to working.
So good afternoon, everyone. I request everyone to please mute. I hope you are able to see my slides. Okay. So a brief introduction about myself. So I am Rishabh and I am a PhD scholar from IIT Kanpur. And today I'll be discussing some concepts about the course cell culture technologies that have been taught in week 1. So that will be in the form of MCQs. So we'll start. So the first question is, first things, let me be clear that this will be an interaction session. So I expect everyone to please bring your questions if you have any doubts, or we have to discuss everything that you have been facing, any problems in the course or something, and as well as in the questions if you are not able to understand anything, so you can ask in between. So starting with the first question, that is PBS stands for. So here are the options. That is fetal bovine serum, phosphate buffered saline, phosphate bovine serum, or pH bound sensor. So sorry for the spelling mistake, but can anybody guess what can be the answer?
Phosphate buffered saline.
Okay. Anybody else? Yes, the answer is phosphate buffered saline. And like, this is the main composition of phosphate buffered saline that we use. It contains NaCl, KCl, and disodium hydrogen phosphate and phosphate dihydrogen potassium dihydrogen phosphate. So this is the basic composition of phosphate buffered saline. And what happens is, why this is the most commonly used buffer that we use in our cell culture system. And anything that we have to resuspend or anything we have to make dilutions of cells, so we make in PBS only. And so it can it has in its name itself, it has saline. So that is NaCl. NaCl is concentrated, saline is concentrated NaCl. So it has more amount of NaCl as compared to the other components. And as I have said before also, that it is a buffer. So buffer components are these two. So these two helps in the buffering action of PBS, and it is considered as an isotonic solution as compared to cells, so that the cells do not burst off when we are making some dilutions for adding into the plate. So these two, the hydrogen phosphates that we have for sodium and potassium. So these helps in the buffering capacity of PBS. And secondly, it is non-toxic to the cells. So this is the main applications of PBS that we use in cell culture. Although it's not a major component, but it's one of the important components that is involved in cell culture. So anybody has any doubt in this question? If
No,
not,
sir.
then we are moving on to the next question. So in which phase of the cell cycle, the cell either progresses towards DNA synthesis and another division cycle or exits the cell cycle to commit the differentiation? So can anybody guess what can be the answer?
G1.
Sorry, can you repeat?
G1.
Okay. Anybody else?
G1.
Okay. Yes, the answer is G1 phase. And what happens in G1 phase basically is that, like, this is the first phase of the cell cycle, in which first thing what happens is it is the deciding point whether the cell will enter into the DNA synthesis phase or it will enter into the dormant phase. So as you can see in the figure itself, so this phase is the G1 phase. Then comes this S phase where DNA replication occurs, and after this comes the G2 phase. So G1 phase, in G1 phase, major proteins and all those cells, all those proteins are made that are involved in the further processing of the cell cycle, that are involved in S phase, as well as those proteins if the cell is going to enter into the G0 phase, that is the dormant phase. So all the proteins that is needed for the cell to remain in the G0 phase also are synthesized in the G1 phase. So anybody has any doubt in this question? So if not, then we'll move on to the next question. So the next question is, which of these features decreases after passage in a cell? So passaging is basically when we just a second.
Length of telomere.
So passaging is basically when we like this is one plate and it is full filled of cells, and then we have to move it to the another plate. So passaging is when we culture these plates, these cells again into another plate. So this is called as passaging in simple terms. So the question says, which of the features decreases after passage in a cell line? So the options are size of the nucleus, size of the mitochondria, cytoplasmic volume, or length of the telomere. So can anybody guess?
Length of the telomere. Length of the telomere.
Okay. Any more answers? Okay. So yes, the correct answer is length of the telomere.
Sir, can you explain how?
Okay. So what happens is, like, you know what's the chromosome, right? So this is basically a chromosome. This is one chromatid. This is the other chromatid. And this is the centromere. So The telomeres are the ends of the chromosome. So these are the ends of the chromosome that are known as telomeres. And what happens is, like, when we look at the molecular level, so these are short ends that are like this. Okay, so this is not a continuous strand, and this is the end of the DNA strand present in the telomere. So whenever the division happens, replication happens, and this region, that is during replication, this region becomes short after, like, during replication, telomerase enzyme is synthesized that helps in the replication of telomere. But during replication, so for example, this is the end of the chromosome. So the telomerase can only replicate till this much of the telomere, and this part leaves out. So after every replication cycle, such part is left out. So that leads to decrease in the length of the telomere. Okay.
So can you
So,
repeat what is passage?
oh, sorry. So this is fine, that how is telomere length decreases.
Yes, sir.
Okay. So passaging is basically when we grow, suppose we took some cells from anywhere. Suppose we thawed the cells from crab preserved vials and took it into the medium. Okay. So after 3 to 4 days, the cells will be completely filled in this plate. Fine. So what we say is that it's 100% confluent. So cells will be filled in this plate. So now we have to move it into the other plate. Okay. So we'll move some of the cells to the other plate and culture them. That some cells will act as an inoculum to the other plate. And while culturing these into the other plate, this is called as the passaging of cells. Okay, so we generally do passaging of these cells. Like, this is the animal cell culture. So we generally see whether the cells have filled the area of the plate or not. Then only we do subculturing, and like subculturing is the other name of passaging. That's it. Okay, so fine. Any doubt in this?
No, sir.
Okay, so then we'll move into the next question. Yeah. So in which phase of the cell cycle, two individual chromatids which make up the chromosome get segregated into each daughter cell? So can anybody guess what can be the answer?
G2 phase.
G2. Okay. Anybody else?
M phase.
Okay. Any other answers? Hmm.
M.
So yes, the correct answer is M phase. That is option B, as you can see in the figure itself. So in, like, I hope everyone knows the basic phases of this M phase. Fine. So like S phase, in S phase, the DNA replicates and two strands of DNAs are formed. So like, and then what happens in G2 phase, like some of the RNA or the major preparation of the division occurs, that is the M phase. And then in the M phase, first phase is prophase. So in prophase, the basic thing that happens is the cell membrane, nuclear membrane is disintegrated, and the centrioles are divided, and they try to reach to the opposite poles, as well as the chromosomes, that the DNA that we have is try like start becoming condensed until we reach late prophase or the transition from prophase to metaphase. All the DNA that is there is present in the form of chromatids. Okay, so in metaphase, all the chromosomes that we have are arranged in the central plate, and bound to the, like, these mitotic spindle that are formed between the centrioles are bound to the centromere, and these hold the chromosomes in such a way that it is arranged in the central. And after this, the centromere or the kinetochore breaks off between two that holds the two chromatids together, and the mitotic spindle tries to move it to the opposite poles. So like this, that happens in the anaphase. And then in the cytokinosis or the telophase that we call it. So in that case, what happens is nuclear membranes start appearing and two forms of nucleus are formed, and that are called as the daughter nucleus, and DNA is unwound. That is, from chromatid, it changes to the DNA unwound form. And after that, cytokinesis happens. So this is the way how DNA is arranged into chromatids and it moves to the daughter nucleus. Okay, so anybody has any doubt in this? If not, then we'll move on to the next question. That is, hepatocytes are cells from. So the options are stomach, brain, liver, or kidneys. So can
Liver.
anybody
Liver.
answer? Yeah,
Liver.
correct answer.
Liver.
It's
Liver.
the
Liver.
liver. So let's move on to the next question. Which of these properties can be determined using cell counter? So exact density of an individual cell surface properties, quantitative growth kinetics, or size of DNA. So can anybody guess?
Option
Sorry, can you repeat?
Option C.
Okay. Any more answers?
Option A.
Option A. Okay. Anybody else?
Option C.
Okay. So the correct answer is option C only. So this is the basic design of hemocytometer, and I'll explain briefly how we do these things, how to measure quantitative growth kinetics. So basically, we'll measure how the cells divide on the basis of their number. Okay, so like this, the first figure that you will see is a hemocytometer. It's a slide kind of thing, and in between there is a region which is marked by the lines that is seen just below this. So these are the lines that are marked, and there are four quadrants. So one is this, second is this, third is this one, and fourth is this one. Okay, so these are the large quadrants that we are talking about, and we count the cells in these quadrants itself. Okay, and each big square contains about 16 small squares. So that helps us to count the number of cells in each big square. Okay, so what we do is we make a dilution. Suppose there are number of cells, and in this we have certain number of cells we have isolated from a plate, and then we make a dilution in PBS itself, so that like it's a 1:10 dilution or something like that. And then from this we take 10 microliter and pour it into the hemocytometer. So this is the other design of, like, from top we can see that how a hemocytometer looks like. And the cells are distributed all over this area, and it looks like this in the lower right corner that is there. So the cells are distributed in this manner, and we generally count the cells that are inside this squares and not touching the surface. Okay, we do not take the cells that are on the surface. So we take cells only that are inside these squares only, inside these big squares. And then what we do is we take a calculation, that is how many cells that we have counted, number of cells, into the dilution that we used. So here it's that dilution factor is 1:10. So into 10 by the number of quadrants that we have. So this gives the number of cells that are present in that particular volume of cells that we have. Okay, So this is how we measure the quantitative growth kinetics of these cells. So we can see that from like we grow the culture about 3 days before, and after 3 days we get this many of cells. So this is how we can correlate with the number of cells using this cell counter. Okay, so anybody has any doubt?
That formula again, you
Sorry,
Okay.
can you?
can you again explain the growth kinetics, the formula of number of cells into
Okay, so growth kinetics, it basically is the same formula that we use for the growth of the cells. That is the finite number of cells that we have. Then we have the initial number of cells that we had and into 2 to the power n. Okay, so n here basically comes from the time that we have upon the doubling time. So this formula is used for calculating the growth kinetics, and this is the technique that we use for counting the number of cells. Okay. So let's move on to the next question. So which of these do not lie under biological safety hazards? That is genetic manipulation, radioisotopes in sterile cabinet, propagation of viruses, and position and maintenance of laminar flow hoods. Can anybody take a guess?
Is it genetic manipulation?
Option A.
Option A. Okay. Anybody else?
Option B.
Option B. Okay. More. So the correct answer is option B, because biological safety hazards include all these things: genetic manipulation, propagation of viruses, as well as the laminar flow hoods that we use for doing all the cultures. So for plant cell culture, animal cell culture, or bacterial cell culture that we use will come under these safety hazards. So the radioisotopes comes under chemical hazards, not in the biological safety hazards. So it will be excluded from this list. Okay. So anybody has any doubt in this? If not, then we'll move on to the next question. So in culture systems, osmolarity of a medium is measured by measuring volume of the solution, measuring pH by titration, or by depression in freezing point. Can anybody take a guess?
Measuring pH?
Measuring pH. Okay. Anybody else?
Measuring.
Measuring volume of the solution.
Okay. Anybody else? Any options, guesses?
Option D.
Option D.
D. Okay. Yes. So the correct option is option D. So actually, freezing point is considered as a colligative property. So what happens is, like when we add a solute in a solvent, okay. So suppose this is the solvent and we add a particular solute to this. So what happens is the molecules inside the solvent increases. So due to this, the freezing point of the solution, that is before the addition of solvent, decreases after the addition of the solute. Okay, so in this case, what happens is the freezing point that was before. So we counted as FP, that is a bit high. Okay, so after adding the solute, the freezing point of the solution will decrease. So that's why like after adding a solute to a solvent, the osmolarity of the solution will increase. Okay, so by increasing the osmolarity, the freezing point of the solution will decrease. So the same thing is being explained in the graph itself. So when we cool any solution, so suppose this is the yellow line that we see, and this is the freezing graph of that solution. So after some time, this freezing point appears. Okay, so but after adding the solute, what happens is this temperature comes down quickly, but the freezing point is a bit reduced. So it is the like if the freezing point is in minus, so it will be towards zero now after adding the solute. So that's why there is a increase in the freezing point. Sorry, decrease in the freezing point. So this is how we check the osmolarity of the medium. Okay. So anybody has any doubt in this? If not, then we'll move on to the next question. So bubble formation is prevented in culture systems because it causes shear stress to cells, changes color of the media, changes temperature of media, or causes contamination. So can anybody take a guess?
Causes
Changing
contamination.
temperature of media.
Okay. Any more guesses?
Causes contamination.
Okay. Any more guesses? Sorry, I can't hear you. Can you bit?
Option A.
Okay.
Chatri.
Okay, so the answer is option A. And how does it happen is like if we suppose grow in any culture system, either it is a plate or a bioreactor. So especially in case of animal cells, what happens is the cells are very fragile due to absence of all the cell wall or the fixed membrane. So cell membrane is present there, but cell wall is not present there. So that makes the cell susceptible to any stress on the cell membrane. So whenever there are bubbles in the system, it causes shear stress of the cell that breaks the cell membrane. Okay, so we prevent the bubbles to be formed inside the culture system of animal cells specifically. Okay, so anybody has any doubt in this question?
If the causes of contamination, it can cause contamination to it because bubble formation will increase the moisture or something like that.
Sorry, can you repeat?
I said it can cause contamination to it because of
Uh,
bubble
actually,
formation.
hmm, actually, bubble formation cannot cause contam because, like, there will be no region or like no entry of any other fluid inside the culture system. Okay, if it's added like for an experiment or something, then it's another case. But generally, we don't do it purposefully. So it will not be a source of contam in that case because there will be some CO2 levels also that will be maintained for growing these cultures. So contamination cannot be the case.
So there is no possibility of airborne contamination.
Sorry, can you repeat?
So there is no possibility of airborne contamination.
There can be, but that will be so minimal for these cases. Okay, because like
Okay.
if I talk about a bioreactor, okay, so we have, for example, a tube, and this is the inlet for CO2. So it will be blocked, like it will, the pipes and all those things will be so much of sterilized, as well as a compact structure will be there, so that no contam will be there other than the CO2 levels that are being plunged inside the bioreactor. Okay, so there is least chances of contamination from this point. So that's why it is not considered as a contamination.
And so one more thing, if there is
Hmm.
a bubble formation, it can
Hmm.
lead to I mean, not a uniform distribution of the nutrient to the cells or the culture we are growing in the fermentation media, fermentation vessel. Is it possible?
Hmm.
And that can lead to change in pH and color of media.
Uh, Actually, change in color of media is due to any contamination. So if any other bacteria or fungal contamination is there in the animal cell culture media, that leads to the change of culture of uh, the color of the media. And secondly, about pH, there is another thing that I'll discuss in later slides, that CO2 also helps in maintaining the pH of the media rather than changing the color of the media. Okay,
Okay.
so it will not lead to change in color of the media. Okay, so let's move on to the next question. So islet cells are derived from, can
Pancreas,
anybody take a guess?
pancreas.
Okay.
Pancreas.
Hmm. Okay. Yes, the answer is pancreas. And like, so this is the basic structure of pancreas that we know. And pancreas has islets of Langerhans, and both, it's actually a both exocrine as well as an endocrine structure. So islets represents the endocrine structure, and it has different cells. For example, there is alpha cells that secretes glucagon. There is beta cells that secretes insulin. And if we have to culture any of these cells, then we'll isolate islets from this, and then we'll culture in the artificial medium. Okay. So let's move on to the next question. So which of the following is not a constituent of animal cell? So this is a simple question.
Cell
Uh,
wall.
Cell wall.
yes, yes. So cell wall is not present in the animal cells. Okay, so this is the basic structure of the animal cell that we know. So it has cell membrane and all the other organelles except chloroplast, as well as cell wall. And many of the functions are different from the plant cell that we'll not discuss in this particular slide, because this is out of context. So let's move on to the next question. So there is a motif called as RGD. So in that G stands for. Can anybody take a guess? Anybody knows about RGD? So he or she can take a guess. Is it glucose,
Where
glutamate?
RGD exactly is
Hmm.
where I can,
Please repeat.
but where exactly RGD is used is, I don't
Oh.
really know what it
Okay. So let's discuss this. So this basically represents glycine, and where it is present is it's a tripeptide. Okay. So it's a tripeptide that is made up of arginine, glycine, and aspartic acid. So, uh, and it is present where it's present, as you can see in this diagram, it's present above the integrin, this RGD peptide, and it helps in the cell adhesion of the cells. So what happens is this is the cell membrane, and these are the integrin molecules, alpha, beta, and there is a small peptide that will be present that helps in the adhesion of cells to the other cells or to the extracellular matrix or anything, like for cell communication also it is used. So this peptide helps in adhesion of cells to the surface of other cells or the extracellular matrix, basically.
We have tight junctions where you talked about alpha and beta. So is it related to that tight junction?
Sorry, can you repeat? I'm not getting.
You mentioned the alpha and beta, so is it concerned to anyway the tight junction present between the cells?
Yes, it's related to tight junction basically. That's why it is like helps in addition of the cells to the extracellular matrix or the other cells that are arranged in a tissue form of a thing.
Okay.
Okay, so now let's move on to the other question. So cardiac cells are mostly characterized as. So can anybody take a guess?
Muscle cells.
Uh,
Option C.
neuronal cells.
Okay. Anybody else?
Option C.
Option C. So yes, these are characterized as muscle cells, and like this is the basic diagram of heart that we talk about. And so from heart, we make it as cardiac muscle. So heart is made up of cardiac muscles. So what the cells that we isolate from a muscle will be cardiomyocytes. So that's why we would call it as a muscle cell. Okay, so we are not going into much detail into this because that's not needed here. Just you guys need to know that heart is made up of muscles, that is cardiac muscle, and it is different from the smooth and skeletal muscle in some sense. And the cells that we have are cardiomyocytes, and it's a muscle cell rather than other cells. Okay, and like somebody also said that it's neuronal cells. It is not a neuronal cell, although there is some electrical activity going on in these cells. That's why it is different from the smooth and the skeletal muscles also.
Okay,
Okay.
sir.
Hmm.
Okay.
Yeah. So cardiac cells are the muscle cells. So let's move on to the next question. So HeLa cell line that we commonly use in our cell culture system, so was derived from which of these cells? Can anybody take a guess?
Cervical cancer cells.
Yes.
Option B.
Hmm.
Cervical cancer.
Yes. Option B, that is cervical cancer cells. And can anybody tell from which person this cell line was extracted?
Andresia legs.
Yes. So she was the person and she was suffering from cervical cancer that was caused from human papilloma virus. And like she was admitted in the Philadelphia hospital, and from there the doctor that was treating her took out some tissues from the cancer cells, and he grew in the culture medium. And this is the basic morphology of HeLa cells that it looks like under microscope. So it has been used widely around the globe for many studies, especially cancer studies in therapeutics and all those stuff. So this is the major cell line that we use in that you'll find in any of the cell culture labs that are working on any disease model. Okay. So let's move on to the next question. Yes. So which of these gases, along with its buffer, is used to maintain pH in cell culture mediums? So can you take
CO2.
CO2.
Yes, CO2 is the answer. And how does it helps in maintaining the pH? Can anybody tell me, or like can anybody guess?
They do not contaminate the culture.
Yeah, that we know, but how they are helping maintaining the pH? That's what I'm asking.
It reacts with water to form organic acid
Yes.
and it lowers the pH in that way.
So yes, this is the main reaction that is helped in maintaining the pH. So what happens is whenever CO2 is drained in of the medium, so water is the main basic solution that is there in the medium. So when carbon dioxide is pumped in, it reacts with water and form carbonic acid, and it dissociates as it is a weak acid. So it will not fully dissociate and release H plus ions, but it will give a basic ion, that is HCO3 minus, that is bicarbonate ion, and H plus. So whenever there is increase in the pH, that is the H plus ion decreases. So this releases H plus ion, and that will lead to the maintenance of the pH. And if H plus ion increases, that is there is lowering in the pH of the medium, then the H plus will bind to this bicarbonate ion and helps in the formation of carbonic acid. So that helps in maintaining the pH, and this is all these things helps in like follows the principle of de Châteaubriand's, that is whenever there is change in the pH, that is increase in H plus ion or decrease in H plus ion, then that thing will move on to stabilizing the pH of the medium using bicarbonate ions. So anybody has any doubt in this mechanism?
No, sir.
Sir, what if the pH gets more acidic? Can it balance then?
Yeah, that's what when the H plus ion increases. So what happens is the like it will bind to the bicarbonate ion and it will form carbonic acid. So this helps in maintaining the pH, and it will uh, maintain the pH as it is. Like it will be around 7 in the cell culture medium. So it will maintain around that. Okay.
Okay, sir.
So let's move on to the next question. So there is a blank space that implies that the synthesis of specific products or other specialized functions are under regulatory control and can be reinduced if the right conditions are provided.
De-differentiation.
So what
Differentiation.
uh like
Option D.
de-differentiation?
De-differentiation.
Okay,
De-differentiation.
okay. Sure, that is de-differentiation.
Differentiation, because
Yes.
we are talking about specialized functions.
Okay, so like the correct answer is de-adaptation instead of de-differentiation. So what happens is in de-differentiation, we are making like the cells into its original form. Like before differentiation, what it is, we are making that. So we are going in a reverse dot. But what is adaptation? Adaptation is basically change in the process that is going on, and it will like it's a normal process that we can change that thing into an or other thing when stress is there. Okay, so it can be understood by this figure itself. So consider this as a normal growth, and during gene expression, there is translation of some proteins. Okay, when there is signaling or there is need of those proteins, then this phosphatase will uh, degrade this phosphate group and make these proteins active. Okay. And similarly, when this protein is needed alone, so there is disassembly of this compact structure and release of the separate proteins. So this is the normal case scenario. Okay. So now in case of stress, what happens is there is degradation of these, not like if these are in active states, the green one. So in stress conditions, these green ones are moved to the inactive state. Okay. So this is basically, and if this stress goes out, then these proteins come back to the normal phase. So this is adaptation, the normal thing, and de-adaptation is when the stress comes and all these proteins moved onto a inactive state. So this is de-adaptation. And after the stress is removed, the normal thing is alone increased, that is the growth phase is alone increased, and that again the original thing that is happening inside the cell is taken over. Yes. Somebody raised a hand. You can ask the question. If anybody has any doubt, you can ask. Yes, many people were comparing it with differentiation and like confused between adaptation and differentiation. So in differentiation, what happens is, let us take an example of beta cells that secretes insulin. Okay, so there was some lineage of the hepatocyte cells, and like earlier they were dividing, and then after some time it becomes the beta cells that are present in the islets of the pancreas, and it secretes insulin. So it is moved to a specific function. Okay, so it is not happening with each of the cell of the body. These are specialized cells that are involved in the secretion of insulin. So if we want to get these cells back to the original state or the precursor cells that were there of these beta cells, then this is called as de-differentiation. Okay, so this scenario occurs only when we provide some growth factors in this. Okay, this thing doesn't apply in normal scenario. In case of adaptation, what happens is, like if we say that this protein secretes a this cell secretes a protein. This is A. Okay, this is a membrane protein, and it helps in normal signaling of the cell. Suppose it helps in the translation, and in any stress condition, what happens? This A protein is increased. So it can be anything, like there is high temperature, more acidity. So this protein synthesis increases above the threshold level. So for example, in the normal condition, 10 proteins were formed of A. And in stress conditions, 1,000 proteins of A are formed. Okay. And now what happens is, like the 10 proteins wala conditions is known as the adaptation of the cell. So in that scenario, only 10 proteins will be formed. And in the stress conditions, 1,000 proteins are formed. So during stress condition, the cell is adapted to produce large number of cells. So is my screen visible? Sorry, there was some interruption in the network.
Yes, sir.
So like if the protein starts producing this 1,000 molecules of A, that means it is adapted to that condition. And when the stress condition is reversed, then the cell can bring back this mm, levels of protein to 10 itself. So after the stress will be removed, the number of proteins that are being formed is 10 in number. So this is de-adaptation. Okay, so if anybody has any doubt, can ask. If not, then we'll move on to the next question. So the question states, what will happen if kidney cells are isolated, separated, and grown in culture? So the options are they will grow and form a liver kind of structure. The cells will grow in culture separately but will not form any of the structures. The cells will have a different morphology as compared to the cells in vivo, that is inside the living system, or both B or C can be correct. Can anybody take a guess?
Both B and C.
Both ways.
Yes. So the answer will be both B and C. And because there is some difference in growing in cells in culture or in vivo. So when we say in vivo, it like, for example, we are talking about kidney cells. So kidney is made up of n number of cells. So i- these are packed to form a compact structure and to have a specialized function. Okay, so they are linked with some proteins, tight junctions, or any of the proteins that are transport proteins that link both of the cells, as well as there are membranes, there are blood vessels that transport the blood or transport materials toward to other cells. There are capillaries. So various gases are there, and when these cells are grown in culture, so we have to separate these cells and these will grow separately. And what will happen, the proteins that are formed in this scenario, that is in the compact structure, will not be formed in this because there is some signaling that happens in these cells that helps in growing those proteins that are present when these cells are compact. And when we grow these cells in culture, they are separate. So there is not that signaling happens. So the particular proteins present inside the kidney to form a compact structure are not there. So this is the major difference between the cells that are in vivo and in vitro. Okay. So anybody has any doubt?
No, sir.
Okay. So then we'll move on to the next question. So you are testing a drug that kills cancer cells using cell culture. So you have to be like analyze this question very carefully. So we are testing a drug that kills the cancer cells using cell culture with the help of a brown compound. So that is used as a tester. So that turns purple if the cells are living. You tested three concentration of the drug, 3, 5, and 10 micromolar, and found that with the increase in concentration of the drug, the intensity of the purple color decreases. So what inference can you dug into with this observation? So there are more number of cells alive in the cell culture with 10 micromolar drug, more number of cells alive in the cell culture with 3 micromolar drug, or there are less number of cells alive in the cell culture with 3 micromolar drug, or none of these cells are alive after administration of the drug. So can anybody take a guess?
Option B.
Okay. Sorry, the answer was visible, but please like try to understand the question and then give the answer.
Sir, it's option B
So
because as
because
the concentration, the purple color is decreasing. That means the living cells are
Hmm.
also decreasing.
Okay. Yes. So the answer is option B. And this like the test I'm talking about in this question is the tetrazolium test. Okay, so what happens in that case? It's a like how high throughput type of test in which we can use a 96 well plate. First seed the cells in each of the wells, like a particular number of cells that we seed in these plates. Then what we do is we do the application of drug into certain wells, and then we grow the cells and like the normal first we allow the drug to take some action, and after some period of time, for example on day 0, we tested the drug, and on day 3 we have to check whether their cells are alive or not. So on day 0 we'll do all the administration, the drug injection and all those things, and then on day 3 we'll do the MTTSA, that is the tetrazolium test. So what we'll do in that case is we'll remove the media and add the brown compound, that is tetrazolium, and that is brown in color when it is not reacting with the components of the cell. And when it reacts with the components, that is the mitochondrial reductase that it has. If the cell will be living, then it will have the mitochondrial reductase that will reduce NADH to NAD plus. So during that point of time, the brown colored compound or yellow colored compound will turn into purple. So the number of cells that will be alive will give the intensity will be directly proportional to the number of cells that are alive. So in now this is the principle of the test that we are using. And now get back to the question. So we used 3, 5, and 10 micromolar concentration of the drug. And as we increase the concentration of the drug, the number of cell kill we would observe is more. Okay, so that is the scenario that is observed in this case. So so this is 3, 5, 10. So as we go on with this, the cell kill increases, but the purple color will decrease because the number of cells alive will be less in number in case of 10 micromolar concentration of the drug. So that's why option B, that is the 3 micromolar concentration, will have more number of alive cells as compared to the 5 and 10. So that's why option B is correct. Okay, so anybody has any doubt in this question and on the principle of the MTTSA?
Can you say the full form of MTT one more time?
Okay, so it's actually I guess some that tetrazolium is the last word. I am not able to recall what is MNT. I'll tell you in the next session after looking into it.
Sir, the color will also depend on the metabolism rate of the cells.
Actually, it might depend, but it will not directly link to the color of the cell because this is a qualitative test. Okay, so either the color will change or not change. That's what we are seeing here.
But we are also seeing the intensity. So
Yes, but in this scenario that we are talking about, the number of cells is not much. Okay, so here
okay.
if the cell is killed, then it means that it is killed and no metabolism will be functioning there. Okay, but if we are talking about any in vivo system, in that case, what happens is there is heterogeneous result. So in that case, we can talk about the metabolism rate of that particular cell. Okay, some of the metabolism will be active during that point of time. So we are not considering that in here in vitro
We are
scenario, but
just
yeah, in vivo
considering the
scenario we can
qualitative,
consider.
if it's living or not.
Yeah. So anybody else? So if not, then we'll move on to the next question. So the properties which make adherent cells different from non-adherent ones are attachment to substrate, encourage dependency, no agitation, and all of the above. You can see the table here
All of
and
the
you can
above.
answer.
All of the above.
Yeah. So yes. So what happens is in case of the adherent culture as well as the non-adherent culture or the suspension culture that we see. So adherent cells stick to the walls of the culture plate that we have, and it will fill up all the plate that we see. So this is the scenario with the adherent cell culture. And in case of the suspension culture, there is no adherence to the surface of the plate, and the cells will be moving freely in the medium. and these cells require constant agitation. Okay, these will die out if there is no agitation taking place in these cells. So this is the basic difference between both the suspension or the adherent cell culture. And like encouraged dependency is seen in only adherent cell culture, not the suspension cell culture, because they are moving in the medium itself. And low and then trypsinization, what happens is when we have to subculture these cells or we have to passage these cells, we have to remove them from one another and from the surface of the plate. So we use a trypsin medium that will help us to isolate these cells from one another, and a single cell will be obtained from that. So we use trypsinization for adherent cell culture, but not for the suspension cell culture. And then for adherent cell culture, we require some uh, treatment to the surface so that the cells can adhere to that surface. Okay, so generally we use polylysine coated plate so that the cells can directly adhere to that surface of the plate. So this is basically the main differences between the suspension and the adherent cell culture. So anybody has any doubt in this case can ask.
Sir, in adherent culture, we to subculture that we use chemical desegregation and mechanical, right? Here we have
Hmm.
talked about trypsinization.
Hmm.
And in the adherent cell, do they create a, I don't recall very well, but do they create a thin film in which, by which they interact with each other, right? In this
Yeah.
adherent, okay.
Actually, they don't directly interact with each other. That is only the like, for example, this is a culture plate that we are growing the cells. And in this case, these cells only look for the surface that is free. So if there is cell growing here, 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 then they'll stop dividing because there is contact inhibition in these cells. So they don't directly like interact with these cells. This is the contact or the mechanical thing that is like inhibiting them to grow further. Sorry. So let's move on to the next question. So cryopreservation is important for preserving cells for a long time. So select the incorrect statement from the following about cryopreservation. So first is the cells are preserved at minus 196 degrees Celsius. Cells are stored in liquid nitrogen. Cells do not require any cryoprotectant for preservation. Or during cryopreservation, the thermodynamic activity of the cells is stopped. So what can be the answer of this? Can anybody guess?
Option C. Option C. Option C.
Okay. Yes, the answer is option C. And can anybody tell what cryoprotectant do we use in our daily cell culture scenario?
EMSO.
Yes. So we
TMS.
use DMSO for as a cryoprotectant, and uh, all of you can see the images, right? So this is the vial for those who have not seen any cell culture scenario in their labs. So in the leftmost figure, we have the vial. Okay, so this is used to storing cells inside the liquid nitrogen. So we have large boxes in case of liquid nitrogen, and we store these vials in these boxes. And this is the container and how we keep the cells inside the liquid nitrogen. Okay, and this is the basic procedure that we do. So before freezing, we add a media, that is a freezing media that provides or that has all the components of the like cryoprotectant and glycerol that we use for keeping the cells intact when they are being freezed, so that the morphology of the cells is maintained in that particular freezing media. Okay, so this is the major step that we follow. Okay, so I hope everybody understood the uh, this particular question. So I am moving on to the next question. So confluency means. increase in the size of the cells, increase in diameter of cells, increase in the number of cells that covers the surface of a culture dish, or increase in the DNA of cells. Can
Let's
anybody
see.
take a guess?
C. Option C.
Option C. Okay. Yes, the correct answer is option C. And here is the figure that shows different confluency of a particular cell. So as we can see here, in the first figure, the cells are not covering most of the area of the layer that we see. So this is around 20 to 30% confluent only. So when we keep this in for incubation for some time, then the number of cells increases, and this is around 40% confluent. And when we keep it for some more time, then they start aggregating and these cells will come close to each other. So now this is around 70 to 80% confluent. And after this, when it fills up whole of the surface, this is 100% confluent. So confluency basically means that the number of cells are there and it covers the surface of this culture dish. And based on the confluency only, we subculture the plates and we count the number of cells, that how many cells are there in that particular scenario. Okay. So has any doubt in this particular question?
Sir, is there a desired confluence for desired cell types, or is
No.
100% always desired?
No, actually, 100% is never desirable, because what happens is when we grow a particular um, cell in a culture plate, so if the full plate is filled with cells, so after some time, if we leave that plate, the cells will be removed from the surface that it is attached. So they start dying out in that condition. So we'll not consider 100% confluency as the ideal scenario in any case. So we generally use the cells when it is 80 to 90% confluent. That is the basic time when we use these cells. So after if we leave after this scenario, the cells will start dying and it will float on the surface of the media.
But are there different desired percentages of confluence for different cell types?
No, I think that will only depend upon the experiment that we have to put on, but it will not depend on the basic cell types. The time that it requires for a particular cell to reach a particular confluency may be different, but the confluency that we use for every cell line is around 80 to 90%, not more than that.
Okay, sir.
Okay, so that's all from my side today. And if if you guys have any doubts from the week one, you can ask.
Sir, actually, I joined a bit late, so can I? Is there any means where I can get the earlier questions?
Okay, so there is the whole recording of this lecture will be uploaded on the site. So you can look at there, as well as the presentation will also be uploaded there. So you can have a look there.
Okay, sir. Yeah.
Okay, so please ask if there is any doubt from the week one of the lectures.
So in the first lecture of week one, we talked about tissue culture and cell culture separately, where we take the explant in the first and we can directly grow, and that is the tissue culture. Or we can, what we say, degrade the cementing material between that and isolate a single cell. So, what is the fundamental or the significance of doing in both ways? Can you elaborate on that?
So you are asking me the significance of the tissue culture or the cell culture?
Yeah, because I somewhere around the line it is very similar, I think. So that
Okay. So basically what happens is when we grow any particular cell in a plate. So tissue culture is like when we have to need some like we need to have some function of these particular cell in vitro. Okay, so we require anything like, for example, liver we are talking about. So we need something of that function only in vitro, then only we do a tissue culture. Generally, we don't do a tissue culture in case of an animal cell, but we do a cell culture instead of animal cell. Okay, so what happens is in case of plant tissue culture, we call it as a plant tissue culture rather than a cell culture. And in case of an animal cell, we call it as a cell culture rather than a tissue culture. So what happens is in case of a plant cell, we extract a tissue that is called as an explant, and we grow cells from that particular tissue itself. But in case of animal cells, we have the ability of cells to grow into like we talk about the stem cells. So in that case, what happens is that can grow in any other cell type. So we don't regularly do tissue culture in that case, but we do cell culture. Okay. And like talking about the function of the tissue culture as well as the cell culture, what happens is when we have to do a particular functional test instead for a particular organ, then only we do a tissue culture in case of animals. And for cell culture, we have like many of the tests for that because there is no direct correlation between the in vitro cell culture and in vivo cells that we have. So that's why we don't like correlate between them.
Oh, okay. And the cell culture we predominantly use in the genetically modified crops.
Hmm. Sorry. Um,
I mean, we use cell culture predominantly in the what we say specifically production of the genetically modified crops?
Yeah. So for the genetically modified crops, also we used tissue culture, plant tissue culture rather. So that's a different scenario that we are talking about. And here we like for the cell culture, what we do is we isolate the cells and then we do genetic modification of that cells, and then either we reintroduce that into particular organism or we generally grow them outside these cells, providing all the conditions that are there. Okay. Anybody else have any other doubts?
I do the cause as precisely terminally differentiated beta neurons or any other thing.
Sorry, can you repeat? It's a bit hazy sounding.
Why we precisely call it as terminally differentiated neurons or be it any other stuff?
Okay, so terminally differentiated means that they'll not be able to get back to the original stem cell type of structure. Okay, so for example, neurons you are talking about. So neurons are dedicated to like have neuronal functions like passing on the signal or producing neurotransmitter, for example. So that they will not be able to go back to the original state. That's why they are terminally differentiated. They will not be able to go back to their original state. That's why terminally differentiated.
Okay.
Uh, am I audible?
Yeah.
So sir, in that case, erythrocytes are also terminally differentiated.
Sorry, can you repeat? A bit hazy sound
Uh,
is coming.
Sir, erythrocytes in that case are also terminally differentiated.
Yeah. In case of erythrocytes that we are talking about, there is a different scenario because the cells uh, do not have their nucleus as they mature. So that's why they cannot go back to their stem cells, hematopoietic stem cells, to again form erythrocytes. So that's why they are also terminally differentiated.
Thank you.
Sure. I don't know if this question is appropriate, but if I talk about the immune cells, specifically B cells or T cells, and if we are trying to conduct an experiment on those cells, like how they react, how they interact with each other. Now, as you already know that for activation of B cells or T cells, they both require each other for each other activation by double signaling, I assume. So how can we is it possible right now that we create that in vivo condition in vitro?
Okay, so in that case, what we need is, for growing these cells, like T cells or B cells, we need some growth factors. Okay, so like for I don't remember the exact growth factor name, but there are some cytokines that are required for the maturation of T cells and B cells. So if we are growing these cells in vitro, we can add those growth factors and continue to grow them. But what is the problem in that case is, like for T and B cells, we cannot grow them indefinitely like these normal cells we grow. We have to grow till a particular extent, then we have to introduce that back into the body. Okay, because then only for a certain extent we have to grow them in vitro. Then we have to reintroduce into like for in vivo thing again. Okay.
So, sir, B and T cells maturation, so this would come under gene therapy, right?
Yeah.
And not totally under animal cell culture.
Yeah, but like for ex vivo gene therapy, we have to extract those cells and we have to grow in culture for some time so that we have the homogeneous cell population for introducing back into the cell. So we have to grow sometime for in vitro, but in that scenario, we have to provide some growth factors also to like culture those cells. Okay, so if there are no more doubts, so we'll close this session for now. Okay, so let's meet next week, next Sunday. Okay. Bye. Thank you guys for being here.
Thank you.
Thank you.
Thank you, sir.
Bye, sir.
Thank you, sir.
Thank you, sir.
Thank you.
Yes, sir.